I'm Dr. Aisha Amreen. In continuity with my lectures of cell signaling, this is lecture number 5 about the calcium signaling. To understand the calcium signaling, first we have to understand about the calcium homeostasis, how the calcium concentration within the cell, within the body, or outside the cell is maintained. There are three ways of changing the cytosolic calcium concentration: by the activating the receptors that serve as the channels, like ligand gated calcium channels, in which the ligand binds to calcium channels and they become open, like in the plasma membrane of the cell, by hormone signaling. Just like before I explained that ligand gated calcium channels, they are open when IP3, a second messenger of phospholipase, as a result of phospholipase C, is produced, that IP3 that binds to the calcium gated channels and opens them. It is one of the example in directly promoting the calcium influx by modulating the membrane potential at the plasma membrane, which in turn opens the voltage gated calcium channels. The other mode to maintain the calcium um, homeostasis is that the voltage gated calcium channels, they are being modulated by the membrane potential, by its active potential or its passive potential. The calcium ions can be mobilized from endoplasmic reticulum and also from mitochondrial parts. So these are three main, you can say, the modes or the actions by which the calcium is maintained. Hormone action through calcium involves the mediation of intracellular targets of calcium ions, which are calcium dependent regulator of phosphodiesterase or kinase activity. So hormone action also involves the maintenance of calcium concentration. Either they split off the phosphate bound to the any enzyme and with the help of phosphodiesterase activity, or calcium mediates the phosphorylation by the help of kinase activity. There are few important channels which are working in the calcium homeostasis. There are four calcium channels mainly which play a critical role in the regulation of the calcium level in the cytosol, which I am going to explain here along with the diagram. Two are highly functional in the basal condition, like sodium and sodium calcium channel in the plasma membrane. Here you can see the sodium calcium exchangers which are working in the plasma membrane. Here you can see influx of sodium and efflux or outflux of calcium channel, which is working to maintain both the ions in the cytosol as well. Next, SERCA in smooth endoplasmic reticulum, calcium channels in smooth endoplasmic reticulum, they are working here and they influx the calcium concentration and pool it in the smooth endoplasmic reticulums. After that, the smooth endoplasmic reticulum, it becomes a storehouse of the calcium ions. So this pump is located on the smooth endoplasmic reticulum. Next, two are activated and used under the hormone or the neurotransmitters action. Two types of calcium channels, which number one is voltage dependent calcium channel in the plasma membrane, like here the calcium channel, and these pumps, they are working in the plasma membrane and depend upon the voltage concentration. Likewise, another receptor, the ryanodine receptor, name given to it in the smooth endoplasmic reticulum, here RYR, it is also working for the efflux of the calcium ions. There are lot many other unipolar or exchangers present, like here in the mitochondria you can see the exchanger is there, import of calcium taking place. So all of these calcium channels, they are either, number one, they are in working in exchange of another ion like calcium sodium exchanger, or they were working in uniport, or working with the movement of calcium ions in one direction. So main function is to maintain the calcium concentration within the cytosol and the concentration outside the plasma membrane. And also they are, all of these receptors and the pumps are, they are maintaining also the concentration of calcium within the smooth endoplasmic reticulum and in the cytosol. This is another explanation of calcium homeostasis, in which you can again watch different calcium ion gated channels, calcium and sodium exchanger channels. Here this is uniport and influx, calcium influx here and efflux of calcium over there. Another thing that we are going to study in detail is that phospholipase C, which producing IP3, is another full cascade which will be discussed later. It also evokes the opening of the calcium channel in the endoplasmic reticulum. So all of these channels, mainly the theme to explain it in the two diagrams is that either influx or efflux, both are working to maintain the calcium homeostasis. Calmodulin. Calmodulin is basically a protein. It is a mediator and its name is attached with the calcium. So it is a calmodulin is a mediator which is a calcium dependent regulatory protein. Its best definition is it is calcium dependent regulatory protein. Around 17 kDa its molecular, and its structure is homologous to the protein of muscle, that is troponin C, in its structure and function. It has 4 calcium binding sites, which calmodulin protein. It has 4 binding sites, and full occupancy of these sites leads to a marked conformational change. Whenever the calmodulin without calcium, it is without bearing any calcium, its structure is different. When the calcium binding sites are open and calcium binds to the calmodulin, the conformational change takes place in calmodulin, and its active sites open up, which activate further enzymes. This conformational change allows calmodulin to activate enzymes and different ion channels. The interaction of calcium with calmodulin is similar to the binding of cyclic AMP to protein kinase. In the same signaling like in the cyclic AMP, which we had explained that after cyclic AMP production, it binds to the protein kinase A, and with the 4 binding sites, it forms the specific conformational chain and then activates further molecules. Likewise, the calcium ions, they bind with the calmodulin at 4 places, and they subsequently activate different molecules. Here, the calmodulin then mediates its action through the CAM kinase. This is the main 3D structure of the calmodulin. The calmodulin structure here into subunits is shown here. When four calcium ions are going to bind at their binding site, the colmodulin acquires such a conformational structure, and it activates CAM kinase, and for a short span the CAM kinase also binds with the colmodulin calcium colmodulin complex, and then CAM kinase is activated. As its name is kinase, so it will further be released for the subsequent action on different proteins to phosphorylate them. Calcium colmodulin diffuses into the cytoplasm, binds to the calcium binds to colmodulin and activates certain protein kinase enzymes. For example, what happens that if a hormone that binds to the G coupled receptor here, and after G coupled receptor binding, G protein is activated, that activates phospholipase C. Phospholipase C action, it's some enzyme that hydrolyzes PIP2 into IP3 and DAG. Here our concern is IP3, so here IP3 is only shown here. IP3 then targets a calcium channel on the smooth endoplasmic reticulum, and whenever the IP3 is going to bind on the calcium channel, it will open, and calcium present in the endoplasmic reticulum will be effluxed. So this is the main function of the IP3 or calcium efflux. While calcium colmodulin protein, it is functioning it is also functioning calcium colmodulin protein functioning in variety of modes. Its main action is shown here. When the phospholipase C, I'm not going again from the start, because G coupled receptor protein is their activation, then phospholipase C, all the story is being repeated many times. So PIP2 after returning after hydrolysis producing DAG and IP3. The IP3 secondary messenger, two messengers, DAG or IP3. We are concerned with the IP3 here. While IP3 is going to open the calcium channel, what happens next? When the calcium ions being effluxed from the endoplasmic reticulum, they run towards the colmodulin and acquire four binding sites of the calcium over here. When the colmodulin that is loaded with the calcium ions, it activates many of the cellular response. Its colmodulin, one of them is CAM kinase, and other enzymes as well. So phospholipase C, whenever it is activated by any of the signal transduction mode, by G proteins or by tyrosine kinase, which is the other type of signaling, as the receptor is different, which will be later on discussed. So main action will be the release of calcium ions from the endoplasmic reticulum with the help of IP3, and the calcium storehouse releases these calcium ions to get the response from the colmodulin. Colmodulin is particularly involved in regulating various kinases and enzymes of cyclic nucleotides generation. Cyclic nucleotide means cyclic AMP, cyclic GMP, and etc., and their degradation. In addition, calcium colmodulin regulates the activity of many structural elements in the cells, like 1. Actin myosin complex of the smooth muscles. You know about actin and myosin. These are the proteins involved in the muscle contraction. Then microfilaments mediated processes, mitosis, granule release, endocytosis, all of them, they are involved in muscular or some kind of contraction and retraction movement. So these all contraction movements or muscular movements, they involve the calcium and calcium or calcium call modeling.